এরপরে তুমি ক্লাসে না আসো
নিশ্চয়ই তোমাদের আগে ওই লোকটাকে তুমি হোয়াটসঅ্যাপ করো, ওই লোকটাকে প্রিন্ট পাঠিয়েছিস, ভালো লাগে লোকটার কাছ থেকে।
হ্যালো,
রিসার্চ না করে প্র্যাকটিক্যাল আছে আমার
এটা দেখতে পাচ্ছো সবাই?
yes sir.
Vectors in biology. Vectors in biotechnology. So in today's class, we will be talking about vectors in biotechnology. And so, what is the definition? Spelling is wrong. So, what are their carrier? Vahak or vehicular DNA molecules which carry gene of interest. So, like a plasmid which carries a gene of interest. A vector, when combined with the gene of interest, a recombinant DNA molecule is obtained. RDT means recombinant DNA technology. Recombinant DNA technology. Acha. The aim of using vector in recombinant DNA is one of the following. Why do you use it? Vector to obtain multiple copies of the gene of interest. For example, cloning vector, which I had already given a brief class about. Then the next thing is to obtain the product of gene of interest. To obtain the product of gene of interest. In this case, we use expression vectors. So expression vectors is, this has got your gene of interest. When you transform it in an organism, it will produce your protein of interest. For example, insulin. Clear? Whenever you have problems, please do tell me, okay? Okay?
Okay sir.
Okay. Then what are the characteristic features? Characteristic features. Ideal vector. It should be able to replicate autonomously. पहले भी पढ़ा है. You should be able to isolate and purify it easily. You can use it for transformation. You can use it for transformation. It should have a suitable marker gene that allows easy detection. It should have the ability to integrate itself or the DNA insert it carries into the genome of cell. It should contain unique target sites. It should contain at least suitable control elements. Example, promoter, operator, and ribosomal binding sites. So these are the characteristics which I had already gone through them. I'm just revising them once more. So generally what the vectors contain? It contains a DNA molecules. And it should, most important thing, it should have an origin of replication and it should have selectable markers, which are 4 types. Selectable markers are antibiotic resistant marker genes. It can be herbicides. It can be metabolic oxotrophic markers. It can be screenable markers. So basically, all the things said and done, antibiotic resistant marker genes, most important ones which are used are ampicillin, tetracycline, neomycin, and kanamycin. These are the most important ones which are used. normally. Herbicide resistance, for example, there is something, a compound known as Basta, huge name, but you keep the name in mind, Basta. Then you have metabolic or for a auxotrophic marker genes, especially they are used in yeast. For example, tryptophan. But when, for example, in the medium, you don't give tryptophan. So in order to have this amino acid tryptophan, the bacteria needs to make this tryptophan. But for example, if this marker is, if this tryptophan is, for example, absent, the genes required for making tryptophan, for example, is not present in the plasmid, then this won't be able to grow in a medium which does not have tryptophan. So it needs to make tryptophan. These to survive in a media minus tryptophan, it needs to have the genes which make tryptophan. And you have screenable marker genes like blue-white selection. Then you can classify vectors like cloning vectors, expression vectors. Gene of interest. In cloning vectors, you try to make multiple copies. In expression vectors, you try to express the genes. Then on the basis of host cell, you have vectors for bacteria. The host is bacteria where you want to transform your vector or plasmid. Then you have vectors or plasmid for yeast. You have vectors for animals. You have vectors for plants. Then cellular nature of vectors, you have prokaryote vectors and eukaryotic vectors. For example, yeast, animal, plants, they are all eukaryotic vectors. You know what are prokaryotic vectors? They are plasmid based and bacteriophage vectors. Plasmid vectors, the important names are PBR32 and PUC vector. They are the most two important vectors. I had already done this class. I want to just explain the different types of vectors. So let us first talk about the plasmid PBR322. This was the first widely used plasmid vector. This is the map of PBR322. It has a small size, 4.36 base pairs. That is 4363 base pairs. 4363 base pairs. It also this vector has a reasonably high copy number. It has got high copy number. High copy number. What happens in high copy number? So once you transform it in a bacteria, it makes many of these plasmids. So the yield of plasmid DNA is much higher. In low copy number, for example, in one may form 4, but in high it may form 40. So it has to have a high copy number. And this P means plasmid. BR, the person who researchers who developed it, Bolivar and Rodriguez. And 322. It is the 322nd plasmid which was developed in that laboratory. You can see it has got an ampicillin resistant and tetracycline resistance, and it has got an origin of replication here. And this is the multiple cloning site between EcoR1 and HIM3. So to construct it, it has got an origin of replication. It has got selectable markers, ampicillin and tetracycline. It has got multiple cloning sites. It is a widely used cloning vector, and it is used as a model system for the study of prokaryotic transcription and translation. Advantages: small size, very easy purification and manipulation. It has got 2 selection markers, so you can get a double whammy, you can give it, like this say. It can be a high copy number, 1000 to 3000 copies can be formed in one bacterial cell. And disadvantage, it has got very high mobility. High mobility means this just the moment you remove ampicillin or tetracycline from the media, it will lose the plasmid. দরকার নেই প্লাজমিডকে তক্ষুনি ছেড়ে দেবে, তাই জন্য এটা ব্যাড, সারাক্ষণ অ্যাম্পিসিলিনের মধ্যে রাখতে হবে. There is a limitation. So limitations in the size of the genes which can be cloned. Suppose the gene of interest is greater than 3 base pairs, then it becomes almost impossible to clone it inside. It becomes almost impossible to clone. Then you have PUC vectors. So these PUC vectors are obtained by modifying the PBR322 vectors. These are smaller than, it's only 2.7 KB. It has got a relatively lower copy number, like that at 1000 to 3000. This has got something like 500 to 600 copies. P is plasmid, UC is University of California, where it was developed. It has got an original warrior application, selectable markers. শুশু পেয়েছে? এই সঙ্গে একটু টয়লেটে নিয়ে যাও যেখানে, যে কোনো একটা জায়গায়। It has got an origin of replication, selectable marker, lacks Z gene, and multiple cloning sites. Acha. Uses of PUC vector. It can be used both as a cloning as well as expression vectors. High copy number 500 to 600 copies. Single easy and single step selection. Just the antibiotic and you can select it. And disadvantages, it cannot accommodate larger than 15 KB se zyada. You cannot put, but this is also quite big. Then you have these plasmid vectors. These are, one second, I have to finish plasmid vectors. So plasmid vectors. So they are based on bacterial F plasmids. They are 40 KB of random genomic DNA. This is a plasmid vectors. Then you have bacteriophage derived vectors. একটু করে এগুলো সম্বন্ধে পড়ে যাবে. They are commonly the phages vector that infect bacteria. They are bacteriophage, like lambda phage vector. It has got a head and tail phage, and you can put something like 49 KB inside. In plasmid vectors, not greater than 15 KB, but here this 49 KB. So in bacteriophage vectors, capacity of insert size has increased. But how do you clone something which is 150 KB long, these genes? For example, many of our mammalian genes, they are so big, huge genes with introns, exons, and everything taking together. Capacity of insert higher. For example, you have these lambda vectors. So this is a picture of a lambda phage. You can see a head region and a tail region, and this is the core region. And what happens? The bacteria is sitting over here. They come and sit down like this, and then they put the plasmid inside. Here it circularizes and integrates, and so this bacteria can carry large inserts. 49 KB long. Disadvantage, it is very difficult to isolate it. Transfection of E. coli is much easier with phage particles. Shelf life, not shelf life, this is shelf life, is infinite. You can keep them as long as possible if you want. For example, if you keep these bacterial plasmids, these bacteria, they, for example, they die, they explode, they get destroyed. in a very long term. You have to keep them glycerol stocks, but then every 2 years you have to rejuvenate these glycerol stocks. There are lots of problems. This is a disadvantage. But here in lambda bacteriophage vectors, this is much simpler. Then the cosmid vectors. It is lambda based vectors. They are hybrid between phage DNA and vector plasmid. So they can act both as plasmid and it can act as lambda phage or bacteriophage. Both it can work. It carries a cos site. The cosmid vector has got a cos site, for example. This cos site is so it also contains cos is for this recombining as a bacteria. These cos sites, they are required for from the bacteriophage. When the DNA gets inside the bacteria, these cos sites are needed for integrating inside the host genome. So therefore, it has a cos site, but it lacks all the lambda genes. So therefore, it will not produce plaques, or plaques means, so in a colony full of bacteria, when the bacteria lies and die, little clear fluid-filled cavities are formed. There you can find your little DNAs, plasmids are found over there. So since it lacks all the lambda genes, when it lacks, it will kill the bacteria and it will not work. Advantage is used to clone genes of interest up to 40 kb. Screening method is very easy. Then you have the phage mid vectors. Here, it is a phage from the M13 bacteriophage. So basically, the important thing is that it can be used to provide single or double-stranded method without any recloning. It has got an origin of replication, lac Z gene, multiple cloning site, origin of replications, and it has got ampicillin resistance gene. Okay. It is 1,500 base pairs long. These are truly plasmid with phage genes. These are linear duplex DNA whose ends are lambda segments that contain all the genes. Both lambda and plasmid function replication are intact. So it's like a cosmid, but it's got that F phasmid part. When it has a combination of bacteriophage as well as it has got a combination of plasmid. Plasmid plus cosmid, you can say. Now let us go to eukaryotic vectors. For example, yeast vectors. Advantage of these eukaryotic vectors is you can put lots of big inserts inside these eukaryotic vectors. They are also known as yeast artificial chromosomes. One of them. Then there are also yeast expression vectors. Then there are yeast cloning vectors. Here you can clone inserts which are huge, several thousands of kbs long. You can clone it inside. Like a chromosome. It is like an artificial chromosome, yeast artificial chromosomes. Like that you have bacterial artificial chromosomes. Then you have animal artificial chromosomes. We have these baculobacter virus vectors. These are animal vectors. Basically, insect. They infect insects. Like they are viruses of insects, these vectors. The virus is a rod shaped with a large double-stranded genome. The gene of interest is expressed during the infection, and very high yields of protein can be achieved by this time of lysis. Then you have the bovine papilloma virus vector, BPV. In humans, you have HPV. So it has got a capsid protein surrounding a circular double-stranded DNA the size of 79 kb. Capsid is something which capsules and encapsulates. 69% of this genome is important for viral function. 31% of this genome can be replaced by any foreign DNA sequence. Example P3.7 LDL. Then you have the SV40 virus-based vector. It is a spherical virus containing double-stranded circular DNA size of 5.2 kb. And the viral protein contains 3 viral coded proteins, out of which large T protein is important for viral DNA replication. Then you have types of SV40. They have got SV40 passive transforming vector, SV40 transducing vector, SV40 plasmid vector. Figure is over here. Please do practice these figures. Then you have plant-based vectors. They are used for making genetically modified crops. They are shuttle vectors. They can grow both in eukaryotic plants as well as in bacteria. That is why it's shuttle vector. Can multiply in 2 different unrelated species. Contain 2 oocytes for replication. One is for say plant, one is for say bacteria. There are 2 types of shuttle vectors, eukaryotic, prokaryotic shuttle vectors. Then you have prokaryotic shuttle vectors. From E. coli, you can infect Bacillus thuringiensis. You can do that. E. coli to B. coli, you can use. Then you have artificial chromosomes. They are synthetically designed DNA molecules. They are assembled in vitro from specific DNA sequence. They act like natural chromosomes. 3 components. A chromosome needs to have a centromere, it needs to have 2 telomeres, and it needs to have origin of replication. এটা ক্রোমোজোমই বানিয়ে দিল। হ্যাঁ, কিন্তু তাদের একটা সেন্ট্রোমিয়ার থাকতে হবে, 2টো টেলোমিয়ার দুদিকে থাকতে হবে, আর অরিজিন অফ রেপ্লিকেশন থাকতে হবে। সো দে উইল গ্রো ইন মেবি অ্যানিমাল সেলসেই গ্রো করতে পারবে। So yeast artificial chromosome is one example. Bacterial artificial chromosome is another example. Then you have human artificial chromosomes. Hack. এই যে একটা ছবি human artificial chromosomes. 47 artificial mini chromosomes. They are used in expression studies as gene transfer vectors. They can be also used, for example, in Huntington's chorea and other type of muscular dystrophy diseases. This gene therapy are done by these human artificial chromosomes. They are used. And P1 derived artificial chromosomes, that is PAC. It is derived from P1 bacteriophage. So this is the end of my class. Any more questions? हाँ, questions है किसी के पास? रुबाब, अतुल?
No, sir.
No,
विजय,
sir.
no. Okay then bye bye. तुम लोग को अटेंडेंस दे दिया, 4 करके सबको अटेंडेंस दे दिया. चलो.
Thank you, sir.
You're
Thank
welcome.
you, sir.
এবার